This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
This is Paul Verschure with the Convergent Science Network podcast, and I'm speaking with Aldo Genovesio, who is here as a speaker of our summer school. And Aldo, you study the prefrontal cortex in the monkey. So what's so interesting about prefrontal cortex? Why are you looking at this structure?
Yeah, the prefrontal cortex is very interesting for many reasons, and my interest in the prefrontal cortex is now related to all the integrative capacity that the prefrontal cortex has, and in the ability of the prefrontal cortex to generate goals based on different type of computations. And I was always interested in the computations that an area like the prefrontal cortex can perform, that are very interesting, and how the information can be combined in the prefrontal cortex in order to reach a goal. And
So the prefrontal cortex in primates is in some sense related to complex behavior, plans, executive control, working memory, attentional selection, integration, associative learning, rule learning. That's quite a list of functions.
Yes.
So are there some underlying dimensions to all these different functions?
I think that what we do in neurophysiology, we try to understand first, the role of the prefrontal cortex for each of these function independently. And after we try to understand if we can explain the same data. For example, this is the case of a working memory. For a long time, we thought that many task activity was related just to a working memory of a stimulus. And after we discovered that a lot of this activity could be explained by just an attentional effect. So we try to understand each of these functions separately, but also together to understand what is really which function come to a play in a specific task. But one common ground could be found in the role of the prefrontal cortex in the generation of goals. And this is a difference that the prefrontal cortex can have also compared to other areas like the parietal cortex that represent space and time, for example, numbers, different magnitudes, but doesn't have this capacity that the prefrontal cortex has to be connected to the premotor cortex in a special way to generate goals.
Okay, so how do you study this ability to generate or maintain goals and strategies in prefrontal cortex?
We designed a task that we called strategy task in order to try to understand how the representation of future and previous goals are represented in the brain. And this task was requiring the monkey to making a choice based on the repetition or not of a cue from the previous trial. And the task was organized in a way in which the monkey could maintain in memory the information about the future goal, and we could study, because there was a delay before the monkey could plan a movement. In this way, we could study, for example, in that period, the neural correlate of maintaining in memory a future goal. So this is an approach to correlate the neural activity with the behavior. You have a monkey that cannot make a choice. You study that you study the neural activity in that moment, and you look at how the information is maintained. And we can study the representation of future goal. If we ask the monkey in the strategy task to remember also what the monkey did before in order to perform the current trial, we can also study what the monkey, what is the representation for a monkey of what the monkey did in the past.
But now, so what you also emphasize there, that monkeys already come to the task pre-equipped, if you want, with some standard strategies that they use in these tasks.
Yes, it was discovered by Betsy Burray in the National Institute of Health, working with Steve Weiss, that when monkey learn to associate set of stimuli to a set of responses, they just they do something more than just learning the association, but they because they understand the underlying structure of the task, they are able to apply two strategies. One we call the repeat state strategy. That every time that the stimulus repeats, the monkey needs to stay with the same response. And the other strategy is a change shift strategy, in which the monkey shift goal every time that the stimulus is different. This is because each stimulus is associated only with one response, so the monkey can apply these two strategies. And they found the lesions of specific areas of the prefrontal cortex is able to damage the use of this strategy. It compromises the use of these strategies while the learning is still going on. So the monkeys are able to learn the association, but they cannot use the strategy. So it's something different, just learning the association, but it's something more of a higher order level.
Right. So, but so now what you found in a subregion of prefrontal cortex, you in substance found cells that are responding to these goals, but also that are responding to these strategies. So what's exactly the kind of physiology that you encountered there?
Yeah, we found a combination of signal at the level of a single cell. This is the power of our methodology of the behavioral neurophysiology, because we can investigate what a neuron does, if the same neuron combines different information or not. And we find neurons that are able to combine a very abstract representation, such as a strategy, with information about the goal, so about what will be the goal that the monkey is going to choose, or with the previous goal. So they encode the conjunction of goal and strategy. And this is also another difference between prefrontal and parietal cortex, the ability of encoding conjunctions of factors, so in this case a strategy or a goal.
Yeah, because in this task, the monkey is trained to, let's say, reach for certain locations or look at certain locations in order to get a reward. And that's also the association it learns, right? There's a cue. The cue is placed in space somewhere. Yeah. And then dependent on the correct response, it will get a juice reward or something of this kind, right? So the goal is now a location in this task space. Yeah. And what I found interesting is indeed you have these conjunctive cells now. So these are cells that would respond to, let's say, the cue, the goal, or maybe the strategy. But you found different combinations here, right? Because some of the cells you measured from in this part of prefrontal cortex were specific to location, to goal.
Exactly.
Right? While others were more mixed in their response. So what's the kind of regularity that you extract from that?
Yes, it's difficult to know how the information combine, not to understand the way, why the information combine in a way rather than another. But we found that the higher order information, the strategy, was not only encoded together with the goal, but also with the stimulus feature. For example, you could have a neuron that was encoding the stimulus A that was leading to a strategy to a repeat state strategy, but not to the change shift strategy. So both stimuli and goals could be associated to strategy in a very particular way. But what I was saying today is that although this is a common rule, we have exception. And when we have an exception, we try to understand why we have an exception. And we found that looking at the data, we found that neurons that were encoding the future goal, what we are going to do next, when we looked at the same neuron, we didn't see a signal related to the previous goal. So these neurons are encoding what you want to do in the future, but they don't know what you did in the past.
Why did you expect that it might reflect your past goal?
This task is a task that requires to the monkey to remember the goal. So we are in a experimental condition where we require the monkey to know the goal. To remember the goal, because based on the previous goal, the monkey will decide the next goal.
But one thing that I don't understand, because on the one hand, it's an association task, right? You get the cue, and now you have to go to a certain location.
Okay, yes.
That you learn these pairs. You're trained, and as a monkey, you're trained for many months on these pairs. Okay?
Yes.
So then how does your previous goal figure into this?
The experiment, in our experiment, we had two tasks. We had one learning task where the monkey were applying also the strategy, and one only strategy task. The strategy task actually didn't ask the monkey to associate stimuli to responses. It was just asking to implement a strategy. So in this case, the monkey was just needed to remember the previous goal in order to choose the next.
Okay.
But without, because today I simplified a little bit the task. But
so there's a dependency then between these goals. Like for
exactly,
instance, if the first goal is left lower corner, then the next one might be right upper corner.
Yeah,
some regularity of that
exactly,
kind.
Depending on the next cue. If the cue is the same, the monkey has to use the repeat stay, otherwise the chain shift.
Exactly. So the cue is then informing the monkey about the rule it should follow.
The fact that it's the same or different.
Right,
yes.
Exactly.
Yeah.
But then, so now here with this task, we understand the manipulation, we understand the role of memory, and now you find two things. On one hand, we have sort of a bag, if you want, a collection of cells with variable responses, right? Some are strategy specific, others are conjunctive. They mix different aspects of the task, but in some sense, they're not reflecting the memory of the task.
Yes.
So that would give you a rather incomplete representation. So in this case, where do you think then the memory of the previous goal resides? Is it still within the system and you were just unlucky, you didn't see it?
No, we found it. We found a signal related to the memory of a previous goal, but it's never that signal is never in the same neuron that represent the signal that represent the future goal. So it is there, the representation of a previous goal, of a previous spatial goal, but in neurons that are different from the neurons that represent the future goal.
And you see this as a principle? That's really an organizational principle?
What I think is that, for example, if you go to buy food, for example, in a grocery store, no, you have a list of objects or food that you need to buy, and for example, you see a milk, you take the milk, this is your future goal, and you put your milk with you, you buy it. And if you see the milk again, you don't buy it again because the milk now, it was a future goal before, but now is a previous goal. So there is the need for us to perform what we call output monitoring function. That is to, in each instant, to know which goal is still pending and which goal is accomplished. Because if a goal is still pending, we need to accomplish it.
Right.
So, and we can do it even in very simple tasks like buying food in a grocery store. But what the results say, showing a separation of representation within the prefrontal cortex between future and previous goal, is that having two separate representation may facilitate an operation of monitoring on these two representations.
Okay.
If we want to know if something was done or not, we look where information it is. So I don't know from the computational perspective how this sounds reasonable, but it looks like that at least in our data we see that we have this distinction between the two, at least in our task.
Right. But then is this representation of the previous goal only pertaining to the previous trial, or does it have a variable depth? Is the monkey also representing the goal 10 trials back?
Okay. I actually I don't think that I never did a very sophisticated analysis of 2 trials back. Maybe there could be an effect, but it was so small to be identified with normal statistical analysis. Maybe at the population level there could have been a small effect, but was nothing visible, let's say. So I think that if there is an effect, it's small, about 2 to 10 trials back.
Do you have the sense that behaviorally the monkey keeps a memory depth that's larger than one trial?
In this task we don't know because of this task. Yeah.
All right.
There are other studies that show that prefrontal cortex can maintain more than one trial in memory, but maybe this trial was not the trial that was asking the prefrontal cortex to maintain for 2 trials. Yeah.
But now the other thing is that of the cells that you measured, you have about, let's say, you then try to classify your different cells, right? So some are conjunctive and some are strategy related and so on. But it's interesting that these classifications stop at around 30%, right? So you always have a large subset of cells that have no interpretation.
Exactly.
So in your mind, what are those cells doing?
Let's say that when I say that 30% of cells are representing the future goal, we had to think that maybe other 10% are representing the previous goal, or another 10% can represent the stimulus feature. So I don't know. I cannot tell you now what is the number of cells that don't represent any variable of a task. But this is an interesting question to know of all the variables that we studied, how many cells were out of a task.
Right.
So I don't know
exactly. No, because there's another aspect to this, right? That in some sense we are interpreting the way in which the monkey brain is describing a task, right? So we say, okay, there are goals, there are cues, there are rules. That's it. That's what we're going to look for. Yeah. But it's not impossible that monkey brain actually is introducing other aspects to a task description that we're not looking for.
Exactly.
So
I agree.
So do you have any idea what these additional factors could be that could help you to interpret these unclassified cells? Yes.
We by chance we looked at the activity, because in the exploration phase of the data we looked at the activity after the delay, after the period of presentation of a stimulus. It could be 1 second, 1 second and a half, and 2 seconds, for example. And we saw that there was a 15% of cells that were modulated by the duration of the previous cue. So that was an irrelevant information, and we were able to look at this because by looking at the raster, we noticed that there was an incredible effect in some cells. So we said, oh, what is this? And we now know that this is an encoding of elapsed time that could not be explained by the reaction time. So we did several analyses to factor out, for example, the reaction time effect. And so we by chance we looked at this cell. So you never know. A cell doesn't do anything because maybe you didn't look at the right variable.
Right, exactly.
So you're right.
So you would say mixed in to these features that are encoded are also temporal aspects of the task.
But I cannot tell you how many of these cells are encoding because this analysis may not have an end. So you never know when to stop to analyze and to look at combination. So at this moment, I don't know if these cells are encoding some other factor.
Very nice. Okay. So now we have these we have an understanding of what this prefrontal cortex does. It represents parts of the task, but also some linking in time, like the previous goal, the current goal, or a possible future goal. And now you interpret these representations in terms of output monitoring.
Yes.
So why do you think output monitoring is a good way to describe these properties?
I think that the separation of goals in the two networks is not output monitoring per se, but can facilitate output monitoring if we assume that some other cell will look at this activity to decide if an external observer that we can think that can be another area or another group of neurons will decide if a goal was accomplished or not, looking simply at the presence of activity in one of these two networks. This is a possibility. Maybe I may be also wrong. But we know that we have failure on monitoring in prefrontal patients. So it's one of the problem of people with prefrontal damage. For example, they are not able to accomplish a series of goals. They get confused. They don't know if they did something or not. So we know that also lesions of the prefrontal cortex or patients with dementia have problems in these tasks. So it's also related to some disease, psychiatric or neurological disease.
So, but monitoring in this case means something very specific, such as goal achievement, or is goal achievement an operationalization to measure something that's broader?
Could be broader. Could be broader. We know that I think that we need to monitor many variables in what we do. For example, maybe we will talk later about my last studies, but I can anticipate that we see that goal is important, and that it's not only in prefrontal cortex important to encode what we are going to do, but it's also important to monitor what we are doing, what we are finishing to do. So goal may be a spatial relevance, not only when we look at the future, but also when we look at what we did And even when this is not important anymore for what we are doing now.
But would you say this frontal area is sort of imposing an intentionality on the world? Because you could argue, look, this frontal area is just filtering everything that this animal is engaged with respect to goals.
Yes.
And just saying, look, okay, which of my goals did I achieve? Where am I failing to achieve a goal? So it's really this massive goal filter. Is that a reasonable way to look at it?
Yeah, I think that there is the goal can be the main function. And I give you another example, and that we have looking at the correlation between neurons. So we recorded several neurons simultaneously. And because we recorded for many, many days and with several electrodes and time, we can have two conditions. One in which we have two future goal cells recorded simultaneously, and another in which we have two previous goal cells recorded simultaneously. And we can look at the correlation between future and future cell and past cell. And we see that they're correlated only the neurons that represent the future goal, but the neurons that represent the previous goal are not correlated. So it's true that prefrontal cortex encode goals, but maybe the future goal has a special relevance that we can see from the correlated activity that can be a way of drive the premotor cortex in a choice.
Right, exactly.
In the selection of one course of action.
So the correlation you observed is only between these goal cells?
The future goal cells.
Only the future goal cells.
Yes.
Okay.
And when we look at the cells that are encoding what you did before, you find by chance the same record in two of these cells, they are not correlated. So they don't have anything. So this is still mysterious for us. We have these results, but we still don't know.
But are you interpreting this like future goals have to be included in monitoring in the future? So I have to sort of maintain these representations and have to make them part of a possible task set or something of that kind? Well, these old goals in some sense don't matter that much anymore. I can slowly forget about them, so I don't have to maintain them.
Maybe they don't have to activate simultaneously other neurons. Maybe they don't need mechanisms of temporal summation, for example, to activate neurons in a competitive way to generate a behavior in the premotor cortex and motor cortex later. So
that means you see these goal cells as also organizers of neural activity. That, for instance, neural activity pertaining to cues that you might see in actions and so on.
Yeah, for now we know about the future goal. We don't know if this correlated activity can apply to other couples or pairs of neurons. We don't know that. We don't know if the dichotomy is between past and future. Now it looks like that, unless we might discover one day there was another reason. But now it looks like it's past future.
Okay, so that means there you have. In the response, the neural response in prefrontal cortex, you find that these future goal cells are tightly coupled in their activity, while past goal cells are not coupled.
Exactly.
And also, does it imply that these future goal cells are also strongly coupled to future cues?
Could be, yeah.
Oh, but there's no data on that.
There is no data, but you are right. Could be, we don't know.
Would you predict that? Would you predict that?
It's difficult to know. It depends on the task. We need to imagine a task to think that.
No, but look, if it's the same task, wouldn't you suggest, look, I have a future goal, then I use that state to also already predict, if that's my goal, then I would expect this cue and I expect whatever, this kind of response.
Because in this task, the goal that the monkey is achieving doesn't predict the future, the next goal after the future. So it's difficult to know if, so it stops there.
Right, I understand. So now we have a bit of an idea about the representation of a task in prefrontal cortex, organized around goals, future and past. Yeah. And then, in some sense, you also followed this notion of a hierarchical structuring of the frontal lobe, and you moved to the frontal pole, which is sort of really the structure all the way.
Yeah, we moved at the end of the brain.
Yeah, exactly. So
there was nothing else.
Beyond that, that's it, right? There's only the skull. Yeah. So, what did you expect to find there?
Yeah, I think that we considering that this area is in a special location that is farther compared to the dorsolateral prefrontal cortex from the premotor cortex, we were thinking, and also considering that this is a primate innovation, is an area that should solve new problems maybe in evolution. I don't know, maybe a trivial idea was to find more complexity that we thought to be more integration of information, like
which also would be consistent, I think, with the literature. No,
exactly,
there's really this idea like there's some hierarchy. It gets more and more abstract, but all the features are sort of abstracted, right?
Exactly. Yes. Okay. And so we found that the frontal cortex had a completely different function. It is a monitoring function. So it has just at least we could study the cells in only a few tasks, but we found that these cells are active only when the monkey receives the feedback about the correctness or not of the behavior, and it represents the goal that the monkey achieved. We don't find any other signal of the signals that I described previously, like the representation of a stimulus, or the representation of a past goal, or a prospective representation of a future goal. Nothing. But when I say nothing, really nothing. No example. So not a few. It was really impossible to find example. And we found this very clear signal in 30% of the cell around the monitoring.
But it's very surprising, right? Because is there anything special about, let's say, the anatomical organization of this structure compared to other parts of prefrontal cortex?
Yeah, if the spatial position is farther from the premotor cortex, and so it speaks more with other prefrontal area than with the premotor cortex compared to the dorsolateral prefrontal cortex.
Right.
So this is kind of spatial, and so it's more distant from the behavior.
Yeah, but this also means this is very odd, right? Because we have this cortical circuit. The different parts of frontal cortex will be tightly interconnected. In this earlier area you measured from closer to the motor cortex itself, you find cells related to future goals, present goal, past goals. So there's a clear temporal window. You would believe, given the anatomy, that this should all percolate up towards
I agree.
the
I
pole.
agree too. Yeah,
And it's not there.
Yeah, it's interesting because these two areas are connected to each other. They have reciprocal progression, but the information that maybe comes up to the frontal pole doesn't activate the frontal pole the way in which we think about. So
but how could you get this selectivity? Because it means the prefrontal cortex deals with monitoring and goals. It operates in a certain temporal window with future and past included. Now you go all the way in the front to the frontal pole, and it lives in the now, right? It's completely just now, completely locked to the task.
Yeah, we have this activity even in the dorsolateral prefrontal cortex, but it's not the only activity that we find.
That's right.
So this is what I was saying, that it looks like a filtering function is applied where everything else is gone and it remains. But actually we need really a model to understand the function, why we need cells like these. Or
But would you say that, does that imply that the brain all the way in the front, it's almost the most exclusive spot of the cortex, is actually completely preoccupied with just assessing how well we do with our real-time performance now? Would you buy that? Would you?
Maybe this is, as we were discussing today, this looks not so important in a simple task like this. In a more complex task, we might understand better the function of these cells. We might think, okay, without these cells, how could we do? But in this task, it is more difficult to understand their role. But what I was thinking is that this signal can help. We don't know why this signal can do that, but can help the transfer of information.
But is this observation consistent with the clinical literature? Like patients with lesions only to the frontal pole?
The problem is that it's never only the frontal pole. The lesions are very broad, especially frontal pole is part of a larger area that is damaged. But so it's very difficult to know now so this is why we study also monkey, because it's neuropsychology in humans is very limitative on that.
Right. But so if we again look at this hierarchy of processing in the frontal lobe, then what you were saying is, okay, areas like orbital frontal cortex, so on, might have a good understanding of the task and the strategies you use, but the only area that actually really know whether you succeeded, really knows success, is the frontal pole.
In a very unique way, yes, that doesn't do anything else, and it has a pure signal, until we find something else. But we'll add to that. But it is that. Orbitofrontal cortex, we studied also orbitofrontal cortex with a second strategy task, more simple, and we found that there is an activity related to the goal at the moment of the feedback, but this activity doesn't depend on the success. So these neurons are just representing the goal as left or right, but they don't consider the success of a task.
Right.
So
but then you see this. So frontal pole monitors now the task and it says success. We did it. And then do you believe it's that information that percolates back into other areas of the frontal cortex to represent the previous goal?
Let's assume that there is already the representation about the previous and the future goal, about the future goal. And this activity about the future goal need to move from a group of neurons to another group of neurons. And let's assume that there is already a connection between the future goal cells and the previous goal cells. But this connection is not enough to activate. This is an hypothesis, not enough to activate the previous goal activity. It may need an additional input. This is a possibility. It may give an additional input that, together with the input from a future goal cell, may activate the previous goal cell. But this is just a scheme just to reason about. But.
Right, exactly. Because the difficulty with that scheme is that in some you really start to think about it as different modules performing specific operations.
Although I don't think that there could be a dissociation. We found future and goal cells in all the penetration. So I don't think about a spatial segregation of these cells.
Right.
So this is not my idea, but there could be because these neurons belong to different population of cells. We know already that there are two different networks, partially overlapped. The overlapping is very small. We have a few hybrid cells. So we have already a segregation in the same area of these neurons.
Right. But would you say there is any critical anatomical difference between the circuits in the frontal lobe and and let's say premotor cortex?
Yeah, premotor cortex has this connection with the motor cortex, so it's very close to the.
But in terms of the local circuit, really how the cells are wired up together.
Within each area, it's difficult to say.
Right.
Yeah.
No, that's interesting, right? Yeah. Because even so, you find these rather variable response patterns in a neural substrate that at the local level is relatively uniform.
Exactly.
Right?
We don't know. Yeah, we don't know what is the difference in activity of cells in different layers.
Right,
this is often a limitation of a neurophysiology that we don't know where we're recording. Often we don't know if it's a pyramidal neuron that send an output outside or an interneuron.
Right,
so
exactly.
We will do that probably there are now new electrodes with multi-contacts and probably this will be the answer to these questions to understand the microcircuit.
Of course. Yeah. So how many cells in the frontal pole could you actually characterize in this way? How
we
big
recorded
a
hundreds, maybe hundreds of cells.
And how many could you classify?
Let's say it was 30%.
So significant. Proportion.
Yeah.
So do you believe that this argues against this very hierarchical view on the frontal lobes and this hierarchical abstracting of information towards the frontal pole?
This very specific signal can be a result of an abstraction, can be something because alpha is a very specific signal, can be the result of an abstraction from many other signals, so in a different way from what we are used to think. So maybe abstraction is not just integration, but is producing something new that could be a monitoring.
Of course, abstraction is also to remove information, right? And to become specific.
Exactly, yes.
Okay.
So maybe that. But for sure we need to study more the frontal pole cortex. We still have this is the first study.
Sure, of course.
Our speculation in the case of the frontal pole cortex is very limited to our results. And this is a difference with other areas where we know we can think and compare the results from different studies and to spend our days comparing.
Exactly.
But here we have only this study and some neuroimaging study where we know that also only a part of the frontal cortex can be an homologous area with the monkey prefrontal cortex.
Right, exactly.
So we have this additional difficulty with the frontal cortex that we don't know exactly that if there is an homology.
Right, exactly. But that's not necessarily bad. That means there's still quite some experiments to be performed.
It's still a frontal pole.
Exactly.
I feel that we are still studying the frontal pole with a common origin from both human and monkey.
So now the other thing is that in this frontal area, the monitoring area, goal-oriented, if you want, the question is, of course, also what's the kind of information it receives from other areas, from other modalities. So actually, if you have a visual cue, what information of the visual cue really enters in this area. And you could imagine that this is, again, maybe symbolic and fairly abstract. And for that, you performed a number of experiments where you start to look at how really very specific aspects of visual stimuli would engage with these decision-making circuits. So what was really the idea there?
Here we use different modalities. We in the frontal pole, we use different cues like orientation, color, and reward. Also, we have this connection with the orbitofrontal cortex, so the number of drops of reward could have been an appropriate cue. But we didn't find any representation of a cue, either as a color, an orientation, or a number of reward. This is very interesting because we found an increase of activity that was selective for the goal in the monitoring phase. It was not just selective for the goal, but was just a ramping up of activity. So without using the as a cue and a reward, we might have thought that that was a reward signal,
right,
with an appetitive meaning. But interesting, when we look at the presentation of a reward as cue, we don't see any change of activity. The activity remains flat. So this is very interesting that the reward per se is not able to activate this neuron. So it is something that has to do with the monitoring of a success independently of getting something good.
Right.
So we were lucky to have, to think about having the reward as a cue.
Of course.
Otherwise we would think about doing that experiment now.
Exactly.
At least one experiment less is done.
Yes, that's very good. No, what I was thinking about is then in the next set of experiments where you sort of backtrack again out of the frontal pole, where you start to look really at this rather puzzling question, right? The responses. So here we have the frontal cortex. It responds to different aspects of a task, but these responses seem normalized in some sense, right? So even though you might have the physical salience of the stimulus vary, it might be small or big, for instance, or close or far, the neural response doesn't seem to be really strongly modulated by these differences in just the visual aspects or the properties of the stimulus.
You're talking about the distance task.
Yeah, exactly.
I showed you cells that are encoding the decision, today. So the decision about which stimulus is farther, which stimulus is longer. And so I focused on this aspect. But there are cells that still encode the feature of a stimulus and the small details of the task.
Yeah, but they don't seem to be strongly modulated by, look, if a stimulus might be more salient because it's nearby or it's bigger, right? The response you find in frontal cortex is not that different as to another stimulus that might be further away and smaller. Or do you find modulations there?
In this case, we have in the distance task we have stimuli that have a different distance from a center, not from the animal. Maybe from the animal could have been different, and we see actually pretty much the same number of cellular encoding with a greater activity. That the first stimulus is farther than the second, or vice versa. So I don't know if you meant that. We don't see an unbalanced representation of looking at the relative representation of which stimulus is farther.
But what you're looking at is really now the physical organization of the display and how this is how this percolates into the neural response in the frontal cortex. And it appears that it seems to be normalized, that these cells actually don't really care about, let's say, saccadic distance or anything like this. It's just say, no, this is a cue, and the cue is in the display, and I have to represent it.
Yeah, right? Yeah. So maybe you want to say that this is what I am presenting is an abstract representation. It doesn't depend on the actual position. So the position can be very different, but the signal is the same.
That's right.
So the important thing is that there is a relationship with one distance
right.
with
well,
another.
You could argue it's like a symbolic representation, right? Because it's not modulated by the analog properties of the cue.
Yeah, but we still have, also in this case, cells with mixture properties. For example, a very interesting cell is a cell that is encoding, for example, that the second stimulus is farther than the first, but only when the second stimulus is on the top.
Okay.
So we have cells that are an intermediate level of abstraction. These are very interesting. These cells that really it depends not only. So this looks like an intermediate representation.
Why are you saying that? You could also argue it's a rule. This cell extracted a rule.
The final one. The final one that represents just which stimulus is farther, independently of the position. It's just the same.
No, or if you say. A certain cue only when another cue is in a certain position. So you have a conditional. Yeah, the response is...
I don't think that these cells represent a rule because the rule is always the same. No? The rule is choose the second stimulus. Choose the stimulus that is farther.
That's your behavioral rule.
Yes.
But you could also argue that the display of the stimuli is following certain rule-like regularities. So for you just described a cell that gave a specific response to a certain cue, but conditional on another cue being in a certain position.
Yes. Yeah.
So I could say that's a rule that defines that little part of the display.
Yeah. Yeah. Okay? You can say that. Yeah. Yeah.
So these
can
are
also
the rules
think in this term.
Exactly. So it's not really feature encoded, but it's rule encoded.
It could be, yeah. You could think, it's conditioned to the appearance of a stimulus in a special location, or you may think that more simply is strictly associated to space and it cannot generalize more than that.
No, right, but
I don't know. It's difficult to know.
But it's an important distinction because in your interpretation, so you're saying, well, prefrontal is not, if you want, really encapsulated and segregated from this dirty, noisy outside world.
It's not just the abstract and the beauty.
Exactly, right? Because it's sort of also the physical properties of the world and of the display percolate into its representation. That's what you were saying. That's where you talked about a mixture.
Yeah, because we know that neurons in prefrontal cortex care about space, they have receptive fields, associated.
Sure.
But some neurons go beyond that. Others, maybe they are representing in a conditional way, as you say, more in a rule-like. But for me, it's more, I see that activity more related to the physical world. But I don't know.
Yeah, but wouldn't this be...
It's difficult to know.
As a physiologist, wouldn't this be, or from a computational point, this would be tremendously annoying because here I have this neuron. It sits all the way behind a huge hierarchy of processing of, in this case, visual stimuli. But where we know we go through areas like if you're temporal cortex, where you have very invariant representations to the world. And now I'm in the frontal cortex, I'm receiving this kind of information. And I can just do my job. So you would believe that, of course, you take the most abstract representation, so you reduce noise and so on, right? So in that sense, in this perspective, you could argue, well, we lose a lot when we drop that very clear segregation between more analog representations of the world. That means they vary with properties of the world.
Yeah, with the distance, in this case,
that are more symbolic.
Would be the distance. But the first level of analysis would be the distance from the center.
Right. But you seem to be much more permissive than I am. You're more like, well, okay, maybe they vary with properties of the world, but doesn't that mean that your whole concept of how this part of the brain is organized and relates to other parts of the brain falls apart in some sense?
So my idea is that we have a basic representation is similar to the parietal cortex of a metric. For example, the distance or if the stimulus is on the top or on the bottom. And after we have a representation that is very abstract. We have something in the middle. And unfortunately, when I looked at these cells that we don't know what they are, it's very difficult to understand based on the temporal profile or their activation what is their role. So it's, I think that also in this case, we need a model to understand, because otherwise it's very difficult to understand, especially why we need certain computation instead of others. So this is my point. Why we need, for example, I'm describing cells that are encoding which stimulus is further based on the order, first or second, but this is not a requirement of the task, because the monkey has just to say the blue or the red is further. So we have, also we were talking at the beginning of the interview about this. Do we have neuron that represent something that the monkey doesn't need to know? The order is something like that, because we could avoid having a representation based on the order, but we still have it. And so it's maybe everything needs to be organized in a ordered way.
Right.
And this is even when the task doesn't require that. So,
but do you think there's something to be gained to try to reinterpret the responses of these mixed cells, as you call them, in a rule, in terms of rules? Would that help you think, to understand how the system works? Or
I don't know.
It's actually a futile exercise.
I don't know. I don't know if it can help to conceptualize that as a rule or as a half a rule, or I don't know. It's very difficult to.
Okay, but now, so the neurons that you looked at, would you say that in the end they operate in a similar encoding space? Like, for instance, they all normalize their responses in some way. That it's like, for instance, if I would have, let's say, a stimulus that's further away or nearby, do I try to normalize all these responses to say, look, no, the only thing that matters really are my decision variables that tell me something about evidence. I don't want to know anything about, let's say, physical organization of the scene. Or do you really see this mixed?
No, I see this mixed.
Yeah?
The cells that I showed you are the pure cells, no?
Okay.
Because otherwise, in a talk, if you start to show a mixed cell, people get confused.
Right.
They say, for example, a common idea is that, we had also problem with some referee in the past, is that how is possible this cell is representing this if this cell is representing also some other variables, but we know that a cell can represent more variables at the same time. But if we present in a talk, for example, a mixture of cell, many people, we need to explain very well
yeah, sure.
what is going on.
Absolutely.
So if we have poor cell and we want to make the point that these neurons make a special computation,
but now, doesn't it raise another problem? Because here we go. Now you allow these mixed cells, so that means these. If I'm reading out a mixed cell, and the mixed cell is active, I have uncertainty because I don't really know. Exactly. Is it the physical display or is it because there's some important decision information?
Yes.
So that would mean that if I have an integration-based model of decision making, which is a popular way to think about it, that you say, okay, I just integrate my evidence over time, and whatever reaches threshold first is what I do. Then your mixed cells would mess up that whole scheme.
Exactly. Yeah. So you need to.
So how are you going to solve that?
You need to have we can only think about a distributed way of representing an information that is mixed to other information and can be still extracted, because all the other information will be filtered out because they would be on the opposite side of, let's say.
Right. But the point is, of course, this is why I was sort of hoping to find some sort of symbolic encoding that you say, look, I just care about informational aspects that pertain to my task, and I'm not biased by salience and so on. Yeah. But now, so that's not what you find, right? And we are already in parts of the prefrontal cortex that are really close to the motor cortex, or premotor, where we're going to execute our actions. So I have to get to, if you want, a pure informational representation in my frontal cortex before I can really make optimal decisions.
Exactly. Yeah.
So where is that happening then?
It could happen in prefrontal, but at a certain time and after a certain computation is performed, we don't know. Also, would be interesting again to look at the correlation of these cells, like we were saying, if the cells that are representing a pure signal are more correlated than the cell. So,
but have you ever found in any of your experiments a majority of responses being driven by these, let's call them pure cells?
No, it's always a minority.
Right.
Yeah, it's always a minority.
No, but this is interesting, right? Yeah. Because this might be telling us that also this idea of just pure rate-based encoding of decision-making might actually not be the reality of the frontal cortex.
Exactly.
Because if you have these mixed encodings with conjunctive cells, further modulated by properties of the display, these responses, these rates of responding are not informative on the goals you have to pursue.
Exactly. So we don't know if the information will be extracted from these cells or if these cells are just an intermediate step for the final computation. So this is what we don't know.
Right. Is the frontal pole better at that?
The frontal pole, for now, we know that there is just one signal. So
right, exactly. So also that it cannot help you,
exactly.
Right?
So it's on the opposite extreme. But also, I was saying today that we don't have to expect every signal present in the prefrontal cortex. For example, in the strategy task, in the dorsolateral prefrontal cortex, the monkey needed to remember the previous stimulus, but we didn't find any evidence in thousands of cells
right, exactly.
of a representation of a previous stimulus. While the new task that I was describing that just require the distance task, just require a monitoring. That doesn't require really a monitoring, but we show that the cells show a monitoring activity. We see that when a stimulus was a goal, it's still represented, but even if it's not necessary to represent it.
Right.
But when it was necessary to represent it, it wasn't.
Right, exactly.
In dorsolateral prefrontal cortex, obviously.
Yes, it is amazing, right? But would you, so the dominant view on decision making in the cortex is the dominant view is very much integration based, right? According to this race model or bounded diffusion models. So your data doesn't really fit that model. So in your mind, therefore, these bounded diffusion models are not an accurate description of this system, or is it just something that you haven't looked at sufficiently yet?
No, I don't think that is in contrast with those models. It's just a more complex task where we can maybe look at a race model where we can study the activity based on a race model, but still we may have competing goals. Like right and left or different objects that compete with each other, even in my task. But the moment in which that happens is very fast. So it's very difficult for us to study, unless we may be studying an interpopulation of cells recorded simultaneously. We may understand more what is going on in the moment of a decision, but the decision is very fast here. So it's very difficult.
But still you talk about what dozens, if not hundreds of milliseconds, right?
Yes. But,
so it's not that fast, is it?
But maybe to understand in our task what a single neuron does, I think that is difficult. Because we don't see the representation of competing alternative, because immediately the monkey can make a decision about one goal compared to the other. So there is no maybe the uncertainty that you can have with a random dot experiment where you can manipulate the uncertainty. Here we cannot manipulate the uncertainty. So it's difficult without manipulating it.
Okay, but wouldn't your data suggest that there must be other modes of integrating information in prefrontal cortex that is more sophisticated than just adding up?
Information like the movement of the dot. So I think that the computation that is required here is not too much. I don't know. I cannot see it in the context of a race model. Right, exactly. So it's more difficult for me to see it within the paradigm.
No, which is good, because I think the race model is not necessarily. It's only the beginning of a story on decision making, and certainly not the end, right? So now, in your last set of experiments, you talked about the encoding of irrelevant information.
Yes, this is.
So why would that be interesting? Because in some sense, it's like, I know these people that you go shopping, you get a shopping list, but first they tell you all the things you do not have to get, which seems very inefficient. So why would you worry about encoding irrelevant information?
Yeah, I think that it's important to encode the relevant goal. We found that mainly we encode the relevant goal because we may find better way of reaching a solution. For example, think about a task in which you need to associate A and B to different positions, but you perform this task, and after a while you understand that you are required only to go right. So you can avoid taking care of or paying attention to the stimulus. Without monitoring, you would continue to perform the task in a more complex way.
Right.
So I think that we have a lot of situations like that where we can find a shortcut.
Okay, so it's like an incremental pruning of information in
Yes,
some
I think
sense.
so. Yeah.
Okay.
Yeah.
So what's the mechanism there? How does this play out in prefrontal cortex?
My study doesn't allow us to understand the way in which this information is used. So we don't have a use of this information. And so it would be nice to design a task where we can see how this information.
But how rapidly does, because here comes the display. I perform my task. Yeah. Okay. So I would expect I have all these cells, I have my conjunctive cells, I have whatever. So all these cells get allocated to describing this task. But now I'm going to figure out that a subset of these descriptions are irrelevant.
Yes.
So then you would expect these cells start to drift again in their responses in some way.
We don't know if these cells were more before. Because we maybe, in learning the paradigm, these cells were double than the cells that we are having now. And now we are just looking at the survivors of a mechanism that is broader than this, with much more neurons involved. And also an interesting point that should be studied is that now we have cells that are encoding the previous goal when it is irrelevant. But what if we start to give wrong messages to the monkey and the monkey gets confused? Will the monkey start to monitor other information different from goal? And will be the dorsolateral prefrontal cortex the right area where to find that information? So I don't know.
Okay.
So there are many questions open.
But you would suggest that there is always a goal-driven monitoring of the task to sort of focus on the relevant information. There's a continuous selection process going on there. Is that what you have in mind?
I think that yes, but not only that, because we have relevant goals that are represented. But the interesting thing is that this representation is not so much bigger than the information about the previous goal that we found when the monkey had the requirement to maintain in memory the goal. That was kind of interesting because we don't find big difference when we need to remember something from when we don't need at all.
Okay.
So that.
So how do you explain that?
It's very difficult to understand that.
Could this be a non-specific kind of memory response? That, let's say, whatever you shoot into this prefrontal cortex, you have a memory field, right? So it will be sort of automatically maintained, non-specifically with respect to the task. Is it
if it's a goal? If it's a goal, yes.
Would you buy that interpretation?
The problem now is that what I think is that now we need to be more cautious when we say that the signal is associated to a behavior. Because, for example, if we can find neurons that encode a previous goal, even when the previous goal was not a requirement. Now, when I have a requirement in a task, I don't know anymore if this is a monitoring activity or a memory activity.
Okay.
As a function of reaching a goal. So now this is my idea that we need to be more. We need to think about this.
Okay. So that means that now, after all these experiments, one important conclusion is that to just only interpret activity in frontal cortex in terms of goal monitoring is not sufficient anymore. There might also be some more non-specific memory effects playing out.
Yes.
This is what you're saying.
We need to. I'm saying that when we know that the task there is a task requirement and we see a representation that we think, okay, we have this representation because there is that task requirement. Maybe it's not because we have the task requirement.
Exactly.
Maybe even without it, we would have that representation.
Exactly right. Yeah. So, but would you have any difference in the physiological signature of these responses, like a non-specific memory response, so that it's not dependent on a specific task requirement, might, for instance, have a different latency, a different amplitude? Are there any differences there that help you?
Because we are comparing different tasks, different monkeys, just a proportion of cells that are selective. So it's very difficult now to say. I would say that the proportion of cells is not so different. And the task is different, has different requirements. So it's very difficult to compare latencies. Would be interesting, I think, to continue this line of research with chronic recording and to see and manipulating the experiment and to understand. This is the way to do instead of doing multiple experiments and comparing them.
So now we know we have sort of different pools of cells, at least around sort of goal monitoring and memory.
Yes.
And so now we have an evolving task, right? So I have one trial, I succeed or not, I go to the next one and so on. What's the information transfer between these trials? What do I carry over in information? Right? These last experiments you described, you try to assess in detail. Look, I have a task configuration. I have a goal. I have an action. I get a reward or not. And now I get my next trial. Right? So the question is, okay, of this trial at T is 1, what's the information that I really carry over to the trial at t plus 10?
Yeah.
Right.
I think that we need to design an experiment to see under which condition we have a memory activity that goes over the monitor activity. It is something more than a monitor.
But I thought you were suggesting today that actually only information on the previous goal really moves over.
So, for example, you can introduce random mistakes. For example, sometimes you don't give a reward to a monkey. And what happened next in the next trial? Is the monkey starting to represent something more than this because it needs to reconsider the rule of a task? And maybe we start to monitor even the characteristics of a second stimulus.
Right.
Yeah. So this would be my, if I could do 10 experiments at the same time, this would be one of them.
So now we looked at prefrontal cortex. So, but in terms of territory, how big a percentage of the neocortex do we call prefrontal cortex?
But we have to distinguish granular and agranular prefrontal cortex, and the granular prefrontal cortex we can study only in primates, and this is a primate innovation. The granular prefrontal cortex, also the rodents have a prefrontal cortex, but it's just a granular. So it depends on what we consider, and but the granular prefrontal cortex probably is. It has a role, an important role in.
But this would be about 1/3 of the cortical sheet in primates.
It could be around maybe 1/3. I can't tell exactly.
So it's quite a chunk. Yeah. It's quite a chunk of cortex. Okay. Yeah. So then the question becomes, how many subdivisions of this should we really consider?
We can consider a lateral part of the prefrontal cortex and a more orbitofrontal part that sometimes is associated to a ventrolateral or to the more medial dorsomedial prefrontal cortex. So it depends on the scheme of connection that you consider, and so the classification is still depends on what you consider important. If it is the connections, to have a common input, a common output, or you look at more at the functions, or different functions of the prefrontal cortex. But
is there consensus on this in the field?
There is consensus about macroscopic divisions. Okay. Yes, but when we look at each microscopic division, you can have more or less subdivision based on how much you want to be specific, how much you want to divide. So in anatomy, we never have a perfect number of areas because also the neurophysiology cannot follow because we just now with neuroimaging, with the possibility to do a magnetic resonance to a monkey, we know more about the location, but it's still difficult to target specifically a part of the orbitofrontal cortex. So when you see, for example, results about orbitofrontal cortex, they don't distinguish too much. All the areas that Carmichael and Price is divided by orbitofrontal cortex. So sometimes we have the tendency to divide more, to be more precise, to see differences, but sometimes when it is too much, we cannot use it to understand the neurophysiology.
Right,
so it's very difficult to understand what is the right level in making divisions.
Right. So now would you say your monkeys do learn this task the same way humans would?
We know that the problem is for a monkey is to understand the rule of the games, as usually. So it's very difficult, to understand, to compare.
Okay.
Because they need to understand at the beginning that touching the screen can produce something. And so it's very difficult also for the monkey to eliminate all the potential alternative to a task. For example, the monkey can focus on the fact that it needs to choose the stimulus on the top always, and after try the stimulus on the bottom, and maybe the monkey find the solution, might go back again to. So it's an exploration of possibilities.
Right.
The training.
So for your task, how long do you train these monkeys usually?
Can be, let's say, rarely less than 6 months, 1 year.
Right.
So I can reach 2 years.
Right.
That's
a lot of time.
Yeah.
So Aldo, to finish up our conversation, there are two things. So you're reworking now on monkey physiology for quite a while, and you have gained also amazing insights in how this system works, despite all the unclarities. But this is also the research aspect of it. So, but now if we would have to if we would like to follow your example in the study of the brain, what would be Aldo's law?
Aldo's law is difficult to define, but I think that what I tried to do was always to be kind of at the frontier of a field, like with the frontal pole, we were the first. With the time, we were among the first. So I like to be one of the first to do something. So that was kind of a rule that is on the other opposite. You may be interested in only one subject and to be a specialist of only one thing. So I chose the other law.
Novelty.
The novelty.
Yes.
I like the novelty and get bored after a while. So I like some new challenge.
Great.
So far was like this.
So Aldo, 5 years from now, I'm going to come visit you wherever you are. Now you're in Rome. And I'm going to confront you with the prediction you're going to make today. So I'm going to ask you, look, Aldo, you predicted X. Did it happen or not? So what's the one prediction you would like to make today that you're most...
I would like to be able to record, let's say, more neurons and more areas together simultaneously to understand really how a circuit made of different areas, like we were saying, the frontal pole, dorsolateral prefrontal cortex work together. So I would like next time to answer more questions. Some of the questions
I don't let you get away so easily. I want a hypothesis on prefrontal cortex.
Yeah, no, I'm very interested in social cognition. So I think that the next step will be talking about novelty. So I'm not continuing studying time, but I'm moving to social interaction. I want to understand really what all these signals, if all these signals that I found previously apply to the representation of a different agent that is interacting with us. So the new challenge is social, I think.
So in 5 years' time, you think you have shown that the prefrontal cortex is the substrate for social cognition?
For some aspects.
Okay.
I hope at least for some aspects.
Great. So Aldo Genovesio, thank you very much for this conversation.
Thank you, Paul. Thank you for inviting me.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.